Good morning. Good morning. All right. So, oh my God, my hair is still having issues. Look at this. Look at this little loop. The girls out there will understand, or the dudes with longer hair. Anyway, all right. So we are going to jump right into prokaryotic gene regulation. So this is chapter 18, and it's a continuation of the PowerPoint that we did last time on prokaryotic genetic variation. All right, let me share. So much sharing. Sharing is caring. All righty. Okay, so we left off on this concept. Let me clear that. On the concept of figuring out how our cells in our bodies that have the exact same DNA have different functions, all right? And it has to do with this concept, right? So if all cells in our bodies have the exact same DNA, let me rephrase. All cells in your body do have the exact same DNA, but they express different genes, all right? So specific genes turn on, and therefore specific proteins are made. So if we look at these two different cells, the first is a liver cell and the second is a neuron cell. They have the exact same DNA. However, they have different genes that have turned on, and meaning they have different genes expressed, all right? So if we look at this, what does that mean to have gene expression looking at this? And what we're really focusing on is this, RNAs being made. So in a liver cell, we have alcohol dehydrogenase gene that has been expressed or turned on. And in the neuron cell over here, we have neurotransmitter genes that have been expressed or turned on. So what do I mean by turning on a gene? So turning on a gene is the same thing as expressing a gene. And what that means is transcription, right? So really what we're talking about is transcription. And we know that transcription can lead to translation. So when we're talking about turning a gene on or expressing a gene, we are taking that gene, turning it into RNA through transcription, and turning that RNA into protein via translation, all right? And just as a little tiny recap, we have transcription occurs in the nucleus, and the main enzyme is RNA polymerase. Translation happens in the cytosol, and the main enzyme is the ribosomes, all right? So this process, the central dogma, is really what we're talking about by turning a gene on. We're expressing RNA and then creating protein. Right? So gene regulation is the process of controlling which genes in a cell's DNA are expressed, meaning which ones are used to make a functional protein. All right. And so we have two different organisms and two different cell types, and they do this in very different ways, right? So we have prokaryotic gene regulation and we have eukaryotic gene regulation. As you can imagine, the eukaryotic gene regulation is very complex, has a lot of different stages where you can regulate gene expression, right? All the way from RNA processing to the transport, to the mRNA, et cetera. Whereas prokaryotic gene regulation really has to occur mostly in that transcriptional regulation. So we're going to see a lot of transcriptional regulation in prokaryotic cells. So prokaryotic gene expression, there's a couple of key points. First, genes are found in operons and are transcribed as a group, right? So genes with a similar function are transcribed all at the same time in a group. And I'll define operon in the next slide, right? So operons and transcribing genes together is really important. We will also see that regulatory proteins will bind to that DNA and either promote or activate transcription, or inhibit or repress transcription. So here's the definition of an operon. And I have had students go through the whole class and then this whole section, and then later on be like, what's an operon? So you have to know this definition, all right? An operon is a cluster of genes that usually have a similar function under the control of a single promoter, right? And remember, a promoter is what will start transcription. So if we have several genes under the control of a single promoter, that means that these genes will be transcribed as a single unit, right? So transcription will, transcription of an operon results in transcription of several genes with a similar or related function, right? So if we look specifically at this cell, we have a prokaryotic cell with its single bacterial chromosome. We look at a specific part of that chromosome and we see an operon. It has one promoter. That is where RNA polymerase will bind, right? Remember, RNA polymerase is going to open the DNA and transcribe, but in prokaryotic cells, you're transcribing several genes. So here we have 1, 2, 3 genes that are all being transcribed together in a single mRNA. So one mRNA is going to have these 3 genes. However, during transcription, you're going to make different proteins. So from this one operon and this one single mRNA, you're going to make 3 independent proteins. They usually have a similar function. All right, so the anatomy of an operon. We have regulatory DNA sequences that are going to control transcription, and we have the coding sequences of the gene, right? So here in red, which I'm indicating, these are the coding sequences for the genes, right? And up here, we are going to have other pieces of DNA that regulate that transcription. So for example, the promoter is a good example of a regulatory DNA where a protein will bind, right? So regulatory proteins bind to regulatory DNA. So proteins bind to this regulatory DNA, and that will cause transcription of our coding sequences of genes, right? As an example, RNA polymerase binding to the promoter, that is a protein binding to regulatory DNA. All right. And these images are from the Khan Academy. They do a great job, I think, of describing these processes. All right. So regulatory proteins can either turn the expression of an operon up, meaning more transcription, or down, meaning less transcription. So let's just look at this top one first, where we are seeing that a repressor is binding to that regulatory DNA. And the regulatory DNA this time is not called a promoter, but it's called an operator, right? An operator. And that's DNA where a repressor is going to bind. All right, so repressor protein, shown here in red, this little repressor protein, is binding to a piece of DNA called the operator, and that fundamentally blocks our RNA polymerase from binding. It can't bind anymore. So therefore, you get no transcription being made. So this is repressing or turning down transcription when a repressor is bound to the DNA, right? On the other hand, if we look at I'll do this in a green, makes a little more sense. So here we're going to look at activators. So an activator will bind to regulatory DNA. So here's our activator binding, right? It's not binding to the operator, it's binding to some other regulatory piece of DNA. And in this case, activators are binding to that DNA, and it is actually increasing transcription. So it promotes RNA pull, and it allows for lots of transcription. So here we are making lots of mRNA, right? So we are increasing transcription. So in this case, activators bind and activate or increase transcription, right? So remember, all proteins, activators, repressors, et cetera, are encoded by genes that have been transcribed into mRNA and made into protein. So I just want to remind you that these proteins, repressors and activators, were genes at one point too. They were coded by the DNA as well. So repressors bind, stop transcription, activators bind and promote transcription. So activators and repressor proteins can be turned on or off by small molecules, all right? And I'm not gonna have you memorize any of this, but more likely, I will give you an example that you've never seen before, and you have to interpret that. And we're gonna go over a POGL that I think is really good at describing this as well, all right? So small molecules can bind to regulatory proteins. So here are our little small molecules, often they're proteins. And they can change their shape and therefore alter their ability to bind DNA. So for example, if we look at this first one here, the activator, this green activator is off. So therefore it cannot bind DNA. If off, no bind DNA, and therefore no activation, and you get no transcription. Right? When this small molecule binds the activator, the activator goes through a conformational shape change, now allowing it to bind DNA. So notice that it now has this shape change, which will bind the DNA. And therefore you can get transcription, right? An example is an activator protein might only become active when it's bound to a certain molecule. Right? And the same thing goes for repressors. So here is where students get often very confused, and we're going to try to go over this in a way that makes it pretty clear. If it's not clear here, you can read the Khan Academy, you can read your book, we can go over a POGL, we can go over it together. So operators may be inducible or repressible, all right? So in the first one, the first example that we're going to look at is called the lac operon. All right. The lac operon is important in making, well, actually, I'm going to put in transcribing genes, important in breaking down lactose, right? Breaking down lactose, all right? So the DNA has promoters, operators, genes. Okay, sorry, got a husband in the background. All right, so the lac operon is normally necessary in transcribing genes important in breaking down lactose, all right? This is a, here, and usually, it has an active repressor. So normally it is off, only turns on, only starts transcribing those genes when lactose is present, right? Because otherwise you're going to waste a bunch of energy making all these genes and proteins, or transcribing these genes and making proteins that are not necessary. It only turns on when lactose is present. right? So it's usually off, only turns on when a small molecule is around, that inducer, all right? So this is an example of, sorry, let me try to figure out. This is the lac operon is an example of an inducible operon, right? And it only turns on when there's a small molecule inducer around. And guess what that is? It's something that looks like lactose, right? So if lactose is there, it is going to kind of bind to that repressor. The repressor is going to come off, and it'll transcribe, right? The trip operon is operon involved in transcribing the genes important in making the tryptophan amino acid, right? It's one of those 20 amino acids, right? This is an example of a repressible operon. And that is because it is normally on, all right? It's normally on, usually on, but it can be turned off when a small molecule called a co-repressor is bound, right? So if trip present turns off because there's no need to make more trip, right? And that's an example of feedback inhibition, right? And we're going to go over this several times. All right, so there's good examples of the lac operon and the trip operon. We're also going to do a POGL of this. So just as again, as a reminder, operons can be inducible or repressible. Two really good examples of that, excuse me, are, So an example of an inducible operon is the lac operon, normally involved in the breakdown of lactose, right? Normally involved in the breakdown. So if lactose is present, lactose will bind to the repressor. The repressor comes off, and now you get transcription, all right? So lac operon transcribes genes involved in lactose breakdown. If lactose is present, you're going to turn on the genes, meaning you are going to increase transcription. We're going to, in other words, remove the repressor because it's normally off, right? The trip operon is called a repressible operon, right? It is normally on, whereas the inducible is normally off, and we have to induce it to come on. The repressible one is normally on. We have to like do something to make it turn off. So this is involved in the synthesis of tryptophan, right? When tryptophan is present, however, we're like, yo, you don't need to make any more, so turn off. So if trip present, we want to turn off genes, meaning we're going to reduce transcription, right? So trip, if it's around, it will bind to the repressor, and it will make the repressor work better, and therefore the repressor will stop transcription. So we're going to reduce transcription by allowing repressor to bind. All right. So in summary, gene expression in prokaryotic cells. So we have these two kinds of operons. The first one is the lac operon. It's an inducible operon. So an inducible operon is usually, it is normally off, right? This is typical in catabolic or breakdown pathways. So you only turn on when when the thing you want to break down is present. So you only want to turn on the lac operon, which is involved in the breakdown of lactose, when lactose is around, right? And you want to break down lactose so we can get some of that energy out of it. All right. The second one is a repressible operon, right? These are normally on, right? This is typical of an anabolic pathway or a buildup pathway, right? So you only want to stop transcription when the thing you are making or building up is around. So if the genes are necessary in making tryptophan and we already have tryptophan, we want to stop it. All right, so that's it for prokaryotic gene expression. And we will get to eukaryotic gene expression on the next part. All right, toodles poodles. Ciao. Hasta la vista, baby. Bye.